Right, I'm going to jump straight in this week into section 9.1.2 of the book, and we're going to discuss installing additional packages for R. Now, this is something we haven't done yet. Everything we've done to date has relied exclusively on all of the native functionality in R. So when you load up RStudio, everything we've used has already come preloaded. We haven't had to install or load any additional packages. However, on this occasion, I've given you a script, a tool called hemotools.r, and that script does depend on two key packages that are not initially loaded into your environment. In fact, they're probably not installed. So we're going to need to take care of that first. And that's really important because in most cases, you're going to be needing to install and use additional packages that you haven't built or written yourself. Someone else has done it for you. That's completely normal, and that's part of coding. You're gonna stand on the shoulders of giants, right? And use stuff that other people have written for you. That's completely acceptable and expected, right? So let's figure that out first. Now, there's a huge caveat here for Mac users. I know from experience that whenever you're trying to do something on a Mac, the architecture of your machine is such that you've got a kind of a different set of rules for doing things. It's really frustrating. I appreciate that, and I haven't got a Mac, so I can't possibly cater for you guys this week. So if you get any problems in doing the following, you are going to need to do one of two things. Either you're going to have to use a university computer, or you're going to have to go and use Posit Cloud. Now I'm going to do a completely separate video for Posit Cloud, so that if you need to use that as a solution, you can, and it won't prevent you from completing your formative and summative tasks this week. Otherwise, we're going to jump straight in here and try and complete these steps in 9.1.2, and then the rest of this section as well, to calculate the average cell count from a large number of hemocytometer images, which I provided. Okay, let's try and get these things installed then. Right, the first thing to do, I've got a completely blank canvas here. I've set up a new R project. I'm going to create a new file. First, it's going to be called hemotools, hemo-tools.r. Okay, and I'm going to leave that blank for now. And I'm also going to create a main.r as well. And again, I'm going to leave that blank. Right, so the next thing I'm going to do is, if I go back to my book here and I navigate to the end of my chapter 9 section, there is a script file that I've made earlier, and it's got everything I'm going to need in order to count cells in images. Now, I don't need you to understand any of this. However, by all means, have a look. It's quite interesting. I wrote it myself. I'm quite proud of this script, believe it or not. And it does something really, really useful, which is to automate the count of cells in images. Let's copy it. Let's paste it into our hemotools file. Okay, and I'm not going to touch it from here on in. I'm going to save it, but I'm not going to touch it from here. Okay. Now the first thing I'm going to do when writing any new scripts to handle these hemocytometer images is I'm going to source that hemotools R script file. Okay, so source hemotools. Of course, you did this last week as well. I'm going to use the function called source, and it's a function, so I need brackets around it, and I also need to put in the file name of the file that I'm sourcing, the script file, in quotation marks. Oops. Okay, and I'm going to run that, Control and Enter. Right, now, here we go. I've spilt some blood. I've got a red error down here. This one's quite descriptive. There is no package called PNG. Okay, what does that mean? Well, I need to use a package called PNG in order to run my hemotools functions. And if you look at hemotools, the first couple of lines here is trying to load a couple of libraries that I need to use. The first is PNG, the second is EBImage. So the first one, resolving this error, is easy for PNG. And I describe how to do it in the book, but you can install that package directly using a terminal command or console command, and the command is install.packages, open bracket, it's a string, so PNG in quotation marks, and I've written that in my console. I haven't written it in my script file, I've written it down the bottom. Hit enter. That will then install the PNG package. That's it, it's done, that quick. And if I source this again, I now get a new error, which is the one telling me that there's no EBImage. Now, instinct would tell me to do this. Okay. However, if I try and install EBImage that way, I will get this warning that EBImage is not available for this version of R. Now, this sort of thing happens all the time. It's very, very frustrating. There are aspects of any coding language which are sort of disparate, and the standard way of installing software for this package is not going to cut the mustard, right? EBImage is actually part of a completely different repository of files and information maintained by something separate to our usual installer, which is called CRAN, right? I forget what that stands for, but most packages are coming from that CRAN source. I don't really care about any of that. I just need to know how to get around this problem. Okay, so I've used this package EBImage in my HemoTools file, so I need to make sure it's installed. How? Right, so this is all in the book. Let's find that out. And it was around here somewhere. Here it is, right? So I am going to have to install another package first called Bioc Manager. Okay, so I'm going to do that, and then I'm going to run this command. Let's do that. Okay, now this one will take a bit of time, I imagine. Oh no, it's done. It's done that. I think it's the second one that takes the time. So once I've run this one, it's all good. I'm going to run this Bioc Manager. So this is kind of another package repository for R. It's you see this all the time where you have to install something a different way. That's it. It's something you have to live with. Incidentally, if you're using something like Python, it's a bit more clear-cut. The way to install Python is a bit more unified. Anyway, I'm not gonna rant about Python now. Right, so it's done. It's successfully installed EBImage. It might take a bit longer on your machines, depending on what you're running. This should also work on the university computers. You'll have to install this on the university systems as well. It won't just be there. But if I run my main script file again now, I shouldn't get any complaint whatsoever. In fact, you can see in your environment on the right, you should now see a number of functions. These are the functions in your HemoTools script file. And the one I really want to use right now is the count viable function. Okay, so I'm going to now move, I'm going to demonstrate the use of this function now, okay, using the example image I've got in the book. Where's that? Let's have a look. Here we go. This is the image that I want to count. I want to count all of the viable cells in this image, and this is a viable cell, and the blue ones are non-viable. Okay, I'm going to right-click, save as, and then I'm going to go hunting for my project, and I'm going to stick it in there. Hemo.png, I've saved it as. Okay, let's go back to our code. Okay, this is the code I'm going to use. This is just to count a single file, okay? But I can show you that working, hopefully. If I'm just going to override everything here now. Right, so this is going to load my HemoTools again. It's going to use that count viable function. If you want to take a look, click on this. And then it's going to store the result into a variable called count. And it's also going to calculate the count per big square. So this is going to count everything in my image. And remember, my image contains all 9 big squares in my hemocytometer. So to get the count per big square, I need to divide it by 9. Let's run this. Let's source this. There we go. I've got a result down the bottom. So it's counted 78 viable cells, and it's divided it by 9 to get 8.67. I know that that's correct. Fantastic. If I take a look at the book again, I know that there are 78 viable cells in this image. Fantastic. Now here's the fun bit. We are going to update our script file to do that for 100 images in one go. And I've specifically given you this Hemo 100 zip. In fact, let's right click and save that link as. Yeah, I'm just going to save it as the zip. So if you haven't come across zip files before, I'm sure most of you have. It's essentially an archive of files. It's a way to package up a lot of files into a single file, which makes it a bit more portable. Now we're going to use R to unzip that. So we're not going to do any file handling here using our Windows Explorer or our Mac Explorer. We're going to let R do all of the heavy lifting here. So don't start extracting things. You don't need to do that, right? Again, if you're using POSIT Clouds to do this, this will be slightly different, so I'm going to make you another video. Otherwise, I'm going to get this extended script here, which is going to handle the zip file this time. It's okay, I've copied that back to my RStudio. You can see that Hemo zip file in there now. I shouldn't be able to open it, actually. Actually, I clicked on it and it does bring it. If you're on Windows, it does actually bring up all of those files. So you can view it, all of the files in that zip file if you want. Let me just take a look. Yeah, here's that slide 24 that I just clicked on. There you go. But you need to count them. Obviously, you don't want to do that. And the whole point here is you're not going to click through each one of these and do it manually. That would be silly, wouldn't it? So let's just overwrite everything with that little bit of script in the next part of the book. Okay, and that's it. 24 lines of fun right here. Let's source this. Right, now, you've got to be patient now, guys. You've got to understand what's going on here. You are doing something pretty amazing. You are counting every single viable cell in every single one of those files. It's created a temporary folder called image files, and it's just disappeared, right? It's finished. So that's also good practice. What the script is doing is it's extracting your zip to a temporary directory. It's looping over every file. It's using that count viable function on every single file, storing it, the results in a large vector, and then unlinking or deleting the folder at the very end just to clean things up. And of course, if you were doing this on a very, very large scale, you would need to delete your files after you process them, because every time you unzip, you're going to take up space on your computer. If you did that for hundreds, thousands, maybe even millions of files, you're going to fill things up pretty quick. So it's good practice just to delete those temporary files once you've done with them. And then all that's happening here is you're taking your results vector and you're printing out the results here. And in fact, you can see this very last line in your output is telling me that the mean number of viable counts in each image is 204.07. Let's just say that out loud again. The mean, or the average number of viable count across all those 100 files in my zip file is 204.07. Unbelievable. If you wanted to see what it was for each file, I could just type out results again. Okay, and that will print out the cell count, the viable cell count in every single image. Okay, I could even do something silly like pull up that on a histogram if I wanted to. I'm not going to do that today. I'm not about visualization today. I'm just trying to get you to run this script, install some packages, and do something super awesome with R. And I hope you will agree that it is awesome. That would have taken you God knows how long, manually, days perhaps. Okay, maybe not days, but certainly many, many hours. Right, so that should help you answer the first formative and summative question on the quiz this week. As ever, if you need any support, get in touch with us via the workshop channel on Microsoft Teams. Better still, come along to the session. There will always be someone there to help you, even after the session has ended. So use that as a resource. And that's it for this video. See you in the next one.